Okay, next I want to get into figure 6, TG upregulation of pulmonary expression in other cell lines. The investigators wanted to look at different cancer cell lines. Here they have MCF, the human breast cancer um, cell line that it has a wild type P53 gene, and also the H1299 human lung cancer gene that also has a null P53 gene. As you can clearly see, pulmonary upregulation from TG is still present. So therefore, it TG upregulation of pulma is an independent function. And this is a northern blot. This shows that the mRNA is present. Here in figure B, uh, you can see the cell lines again, and they look at the wild type and the null P53 gene exclusively, and we can continuously see that TG still upregulates pulma as well. And in figure C, they tried to apply a topside, which is known to upregulate pulma expression in a dependent manner. And you can clearly see here, pulma expression was only seen in the wild type, but not in the null P53. So this is an indicator that TG specificity um, it has specificity when upregulating puma. Next, they want to try to look at isogenic cell lines to basically see if the null and the wild type, the isogenic cell lines, they only differ in the P53, and we clearly see that you still have the upregulation of puma in both of these. Next, they did a northern blot analysis because we already knew that TG had upregulated puma and other cells. So, with the northern blot analysis, they did a double knockout. They looked at the P73, which is a homolog to P53, and is known for the regulation of the P53 gene. Um, and it's also P73 is also known to upregulate puma. They wanted to test to see if P53 null could be upregulated from puma via P73. So this is the reason why they used the S SAOS2 cells. As we can clearly see here, even at the various concentrations, that puma upregulation is still seen in this cell line as well. Next, they want to try to promote, since TG was known to increase uh, calcium concentration by depleting the endoplasmic reticulum calcium pools. They wanted to apply another calcium, calcium promoting intercellular agent, um, N-omycin, and you clearly see that puma is also upregulated with N-omycin. So you can look at this. It says puma is a marker for any changes in calcium concentration, and it's not just limited to TG. Figure 8, they want to do evaluation of TG and the regulations of puma promoter expression. So with the hair, they wanted to look at the promoter regions of TG, and they see at different transcription factors. They wanted to evaluate whether TG still have expression of puma, or whether it's going to downregulate puma, or the TG have any. Could they basically knock out TG? And basically, what they see here in these two lines, that TG is still, you still have activity Even in these regions that they clipped, they still have promoter expression. So we can in conclusion we can say that TG upregulates PUMA and the P53 and P73 genes in an independent manner, and PUMA appears to play a role in TG mediated apoptosis to store calcium in ER and induce apoptosis. The BAX deficient cells have a greater resistance to TG mediated apoptosis, and PUMA also mediates cytochrome C release in the presence of TG. And this will conclude our presentation. Thank you.